I've only explored a very small sliver of what science has to offer. One of my fondest memories is going to the tech museum with my father. There were like some really cool exhibits which I can still remember. Namely, one of them was the Body Worlds exhibit, where they actually had these muscles and they had bones which you could visually see, and that was one of the things which I still remember as one of the coolest instances where I got to see science and I got to see the human body firsthand. My project was focused on G-protein coupled receptors, and to give you an overview as to how important it is to study these receptors, currently more than 50% of human therapeutic markets focus on trying to understand and design drugs to target these receptors, which make them incredibly important for us to try to deregulate or regulate the expression of some chemical within the cell. And due to the increasingly big focus on targeted sort of medications, it becomes increasingly important for us to understand the structures of these G-protein coupled receptors, because targeting these receptors will allow us to target the chemicals in cells, which can allow us to change the way that diseases operate within a cell. So, first of all, what is a G-protein coupled receptor? To draw a structure and to make you familiar with the structure, you can think of it as there's three individual parts. There is an alpha helical part, then there are loops that go from one receptor to the next, and there are 7 alpha helices within the cell, within the G-protein coupled receptor structure. And the brunt of my efforts were focused on trying to study or trying to predict the configurations or the different conformations of the alpha helical portion of the GPCR, because this is the part with the most variation and is the part known to have the most integral function in the GPCR, as it can interact with the ligand within the cell. So essentially, what I tried to do was I tried to predict an example G-protein coupled receptor and its configuration when it's bound to some sort of a ligand. So if you can imagine this being a ligand, I try to predict the conformation of how the ligand binds to the protein structure. And this is originally a very daunting problem, because you have multiple structural issues, you have protein structural characteristics, and you have other issues to deal with. For instance, what direction should each of the helices rotate in? Because I'm trying to design these receptors or trying to predict their structures from scratch. Don't take your ideas, don't belittle your own ideas. And what I mean by this is, whenever I read something, and even when the idea seemed very enormous and there seemed to be a lot of problems which I could immediately think of when exploring an area of research, when I stuck to each idea and continued to read up and edit proposals, etc., it led to positive results, or it led to me becoming more intelligent in terms of tailoring my proposals and my computational studies to fit my needs. So read quite a bit, become very well informed in your area of research, and make sure you continually update your understanding in an area of research, and don't give up on your ideas.